In this video, we're going to go into the topic of amino acid metabolism. It's a really big topic, so we're just going to skim the surface in this short video, but hopefully it'll give you a flavor for what we're thinking about when we're thinking about both how amino acids are synthesized and how they're broken down. So, amino acid metabolism. And as I said, we're going to talk about how they're made and how they're broken down. So first, how do we make amino acids? So amino acid biosynthetic pathways are basically all rooted in central carbon metabolism. And this summary chart here, this is basically showing where all of the amino acids come from and how that's related to things like the glycolysis gluconeogenesis arm of things, to the citric acid cycle, for example. And hopefully what you can appreciate here is there are some central connecting nodes from which amino acids branch off. So a really big one is pyruvate. Pyruvate is the source of the amino acids isoleucine, leucine, alanine, and valine. If you go into the citric acid cycle, you can see that, oxaloacetate leads to aspartate, threonine, lysine, methionine, and asparagine, right? So if we know something about central carbon metabolism, we can start thinking about how we get to amino acids just based on their proximity to different points in the central carbon metabolism pathway. Now, what I was showing there was actually the biosynthetic pathway sort of writ large across all of life. It turns out that we can actually only make 10 of the 20 amino acids. So there is a class of essential amino acids. These are the ones that we actually have to take in from our diet. And then there are the essential ones, or non-essential ones, sorry, which is the ones that we actually synthesize ourselves. And so if you look at these two lists, maybe you can start trying to pick out patterns, what makes them similar, what makes them not. But we're going to talk about where this pattern comes from in just a second. As a really quick aside, there's actually this sort of middle category too. So, for example, the amino acid glycine is non-essential, but some people consider it sort of essential or quasi-essential because it's difficult for our metabolic pathways to keep up with it. So, in general, we want to have more from our diet than we would be able to make ourselves. So, okay, but we can only make 10 of the 20. Why? Well, one pattern we can look for is actually ask how many steps there are metabolically to make these different amino acids. And so what I'm showing here is a graph that I made of each of the amino acids along this axis, and then the number of synthetic steps away from a central carbon intermediate that's required to get there. And then I've colored them orange or blue based on whether they are non-essential in orange or whether they are essential in blue. And one of the things that immediately pops up, at least in my mind, is most of the non-essential amino acids, the ones that we can make ourselves, are over here on the left. And most of the essential amino acids, the ones that we have to take in, are over here on the right. The only exception that we really see here is the amino acid tyrosine, and it's kind of an exception for a reason. So we'll get into that in a moment. But basically, the non-essential amino acids are metabolically close to central carbon metabolites. So let's actually look at some of these. First of all, we have here, it's kind of small, let's zoom in. We're going to fill out this whole page. But so let's look here. This is pyruvate. Pyruvate can be converted to an alanine. And this can be done by a transamination reaction, right? So I'm not showing the partner here, but if we had a glutamine, for example, come in, it can drop off its nitrogen here and swap it at this keto group, and it would come out as a glutamate, and we would now have an alanine. So it's a single step to go from pyruvate to alanine. If we look at, say, another one here, I'm going to zoom again. Now we've got oxaloacetate here. I guess I didn't mention pyruvate. That's the base of glycolysis, right? So it's a central carbon intermediate. Oxaloacetate, important intermediate in the citric acid cycle. And it can be converted to aspartate by a transamination reaction. And then aspartate can be made into asparagine by an amination reaction. So one step and two steps to aspartate and asparagine. Zoom out again. Alpha-ketoglutarate can be converted into glutamate and glutamine by the same basic logic as aspartate and asparagine. And I mean, when you look at this list, we've got 5 amino acids that are all synthesized by a few steps away from these central carbon intermediates. And when you also look at these set of amino acids, one of the things you might notice is if you watched my video introducing nitrogen metabolism, these are kind of big players, right? Glutamate and glutamine are two of the big nitrogen donors that we have in our cells. Alanine is one of the primary amino acids that we use to actually transfer nitrogen in the bloodstream. And so an aspartate is actually at a very high concentration in cells as well, that behaves similarly to glutamate. And this isn't by accident, right? The reason that these can be really good sort of nitrogen carriers, nitrogen currencies, is because they're really close to central carbon intermediates. You can make a ton of alpha ketoglutarate, stick a nitrogen on, and voila, you have your nitrogen currency. So these are our central amino acids to our metabolism, and they're also central because they're close to central carbon intermediates. Okay, let's go on and hit the other non-essential amino acids. So those are sort of the big five. Here's proline. Now proline, we actually have to start doing some work. So zoom in, we start from a glutamate, and then we go through 1, 2, 3, 4 steps to get there. You'll notice I'm not drawing all the other molecules that are involved, but we actually have an ATP that's being invested in this process. So there's multiple other steps involved. But just in terms of number of steps, we are 1, 2, 3, 4, 5 steps away from a central carbon intermediate to make proline. Then there's a whole other series of amino acids that instead of being derived from pyruvate, oxaloacetate, or alpha-ketoglutarate, this one is involved from 3-phosphoglycerate, which, again, if you have glycolysis in your head, this is the intermediate right before the formation of 1,3-bisphosphoglycerate in the first ATP yielding step. So this is an important part of glycolysis. If you look at this process here, what you can see is we start with 3PG, and from 3PG, we can actually go and form, there we go, zooming all the way up, we can go and we can form serine, glycine, and cysteine, right? So 1, 2, 3, there's 3 steps to serine, and 4 steps to glycine, and 5 steps to cysteine. And again, there's a lot of details that I'm skimming over here because there's a lot of complexity in these pathways overall. But hopefully you can get a gist for it, that relatively few steps away from 3PG is say, serine. Okay, cool. Last one we're going to add on here is tyrosine. And if you recall, tyrosine was the weirdo, right? If you think about synthesizing tyrosine from scratch, it's actually a really complicated pathway that involves like 10 steps. We don't have to make it though, or we can make it though, because what we actually do is we take phenylalanine, a essential amino acid that we must consume, and we turn it into tyrosine by sticking a hydroxyl onto it. So it's kind of a cheater non-essential amino acids, right? As long as we're getting all of the essential amino acids, we can make tyrosine from phenylalanine, but we really can't make it from scratch. So if we go back to our overall pattern, it's basically that the essential amino acids are all easy for us to make. We do anywhere between 1 and 5 steps. The ones that take 20 steps, well, we just steal them from other organisms. So, and this is actually a broader pattern across metabolism, which maybe in future videos I'll get into this. But if you say, look at a stationary organism like a plant, or you look at a microbial community, they often invest a lot in exotic chemistry. And part of the reason they do that is because they can't run away, they can't chase things. Whereas critters like us that can move around, we tend to start investing more in things like, building muscles and bones and everything. And we tend to scrap all this complicated chemistry because we outsource that to microbial communities or to our diets. So, okay, but that's an aside here. Overall here, the idea is that the essential amino acids are hard to make, so we steal them from other organisms. Now, I wanted to give a brief I don't want to go through all of them because these are really complicated and it just ends up being a list of pathways. But I do want to just look at a couple of interesting highlights that illustrate points across amino acid anabolism, there we go, from a variety of different creatures. So for example, here's pyruvate. That's one of our central carbon intermediates. And if you look, you're like, oh, look, we can get to leucine. 1, 2, 3, 4, 5. Oops, sorry, that's isoleucine. I wanted to go isoleucine first. 1, 2, 3, 4, 5. We can go to 5 steps. We can get to an isoleucine, which might seem like, yeah, that's something that we should be able to do. That shouldn't be too hard. But then it's 1, 2, 3, 4, 5, 6, 7, 8 steps out to leucine, and there's only one step that's shared. So we have these branching pathways, but the chemistry is actually sometimes not necessarily intuitive. Why do we have, isoleucine and leucine are very similar in some sense, but they actually have very different biosynthetic pathways because of the different starting state intermediates. This very early intermediate, this very early split into these alpha-aceto-alpha hydroxybutyrate versus the alpha-acetolactate sets which sort of outcomes you can get. Okay, so cool. That's a moderately not that complicated, but somewhat complicated amino acid. If we, let's say, look at lysine, I'm going to hide now because there's lots of stuff on this slide. If we go to lysine, what we see is it's 8 steps away from alpha-ketoglutarate. And we might say, oh, it's not too bad. It only takes 1 ATP and 1 acetyl-CoA. But then if you start looking into the pathway itself, what you'll actually see is, like over here, for example, hey, look, we had to do an oxidation, right? We took an NAD plus and it made an NADH, so we oxidized something. They're like, cool. And then you look over here and you're like, oh, well, here, look what we're doing. We're actually doing an NADPH, and then we're reducing something, and then we're reducing something here, and then we're oxidizing something here, right? So the net reaction of 1 step and 1 ATP actually hides a whole bunch of complexity. There's lots of redox chemistry that's being thrown in here. Here, zoom in on that, we have transamination reactions happening, right? There's lots of crazy stuff that happens under the hood to make a lysine. And so this maybe makes sense. This is the sort of pathway we're like, well, we get plenty of lysine from our diets. Why would we need to build all the machinery to make those? Okay, last weird pathway, and then I'm going to talk about some general patterns. And I'm not even going to show the whole pathway. I just wanted to show one part of it. So histidine biosynthesis, it uses ATP. Cool, lots of pathways use ATP. And then this is a molecule called R5 PRPP, which is important for nucleic acid synthesis. And you're like, cool, what's it going to do? Well, it turns out it doesn't do ATP like normal ATP, right? Normal ATP would be you chop, and zoom in here, would be you chop off some phosphates, you extract some energy, you turn it back into energy currency. No, this PRPP is actually covalently attached to the ATP molecule, releasing 2 inorganic phosphates, 2 pyrophosphate that can go off and be cleaved. And then the ring actually opens, and then the base cleaves to make the characteristic histidine ring. So we have our histidine ring over here. And then we made this crazy molecule called ARCAR over here, which is like a chunk of an ATP, right? We literally cut the ATP into pieces. And this is just to show, man, some of these amino acids involve some pretty crazy chemistry to make them. It takes, I believe, the total count, kind of depends where you start from, but it's at 20 steps to make a histidine from scratch, right? So lots and lots of time, energy, and materials get invested by microorganisms, particularly in making these amino acids. Okay, so cool. I just said lots of words about complicated, cool things, but let's talk about some overall patterns. First of all, is we do see lots of branching patterns. So when you think about basically all of the pathways that I talked about, you start with some amino acids precursor here that gets made into another precursor. Then we branch off and we make a different amino acid. So we saw this, for example, when we were looking at 3PG, it went off to form cysteine on one branch, and it went to form serine on another branch, right? So there is this branching pattern of increasing complexity. Second is that the major central carbon intermediate precursors, there's actually a fairly small number of them, but they're oxaloacetate, pyruvate, alpha-ketoglutarate, 3PG, PEP, and E4P. That one's out of the pentose phosphate pathway and goes on to make aromatic amino acids. And then finally, if you started looking at those pathways in detail, what you'd actually see is that amino acids often use other amino acids in the process. So often glutamine, but sometimes glutamate, sometimes aspartate. And that's a nitrogen source. That's where we're getting the nitrogens to build these complicated molecules. So, okay. That is kind of what I wanted to say about the synthesis of amino acids. Now let's talk about the breakdown side. How are amino acids broken down? How are they catabolized? And so zooming out a little bit, we prefer burning carbohydrates and fats for energy. So as a whole, we do. But we will use amino acids if needed. So if we're under starvation conditions, for example, or we have high excess of protein in our diet. And so we will burn that. And that process of burning or of breaking down amino acids means we can also stop and take amino acids and take the carbon backbones and actually turn them into carbohydrates and fats. So there's a biosynthetic route between amino acids and between carbohydrates and fats. When we think about the breakdown of amino acids, there's a high level, there's two parts. There's an amino side, right, of the amino acid, and then there's going to be some carbon bits, mostly carbon bits at least. And it turns out that these two are split and undergo different metabolic fates. So we'll see this in a second, but the ammonia part of the backbone goes into the urea cycle, whereas carbons actually get thrown right back into central carbon metabolism. So, suddenly you've got, I don't know, an alpha ketoglutarate again. And that's just, yeah, so that's what we're going to go through here. And so what we're going to go through is the nitrogen side and then the carbon side. So the big problem that we face with nitrogen is actually that this free ammonia is toxic, and it's toxic for a variety of reasons. It is extremely basic and reactive. It can also bioaccumulate. So there are situations where, for example, it's in the NH3 form. So it's neutral and it can diffuse across the bilayer. But if the pH of the environment inside, say a neuron, is slightly different, slightly higher, what you'll get is slightly lower. It'll pick up its proton and become more positive. So you actually have an accumulation. So ammonia is something we really don't want to have around. If you've ever, I don't know, been cleaning in a small place with an ammonia-based cleaner and you spray it and you sort of inhale it, you know how awful ammonia can be as a sort of acute experience. And so we have this whole thing called the urea cycle, which detoxifies ammonia. And I'm not going to go into it in too much detail because it's a fairly complicated thing, but I'll highlight the investments and the outputs and then highlight one bit at the very top. First, we invest in ammonia. That's the thing we're trying to get rid of, so I guess it's a good investment. A bunch of ATPs, an aspartate, and this HCO3 minus. And what we get out the other end is urea and fumarate. And the rest of this big complicated thing I'm showing over here is a cycle that cancels out basically in that process. The one thing I do want to point out at the very top is this transamination reaction that happens at the very beginning. So what we're going to do is actually take a glutamate and we're going to rip its ammonia group off as part of loading it into the urea cycle. And the way that we do that is by doing transamination reactions where alpha ketoglutarate comes in and picks up the ammonia from generic amino acids. So this is the amino acid you're trying to break down. That's the ammonium or ammonia eventually that you're going to try to detoxify. The step here is sort of generically transfer that ammonia onto alpha ketoglutarate and then onto glutamate. And once you have glutamate, then you have this fairly complicated enzyme, glutamate dehydrogenase. It's actually going to do an oxidative deamination, where it's going to reduce an NADPH in the process to make this ammonia. And so you can basically have generic transamination happen to put it onto a specific carrier. Glutamate is a specific carrier that can be recognized by this complicated enzyme to then donate ammonia into the urea cycle. So I'm just highlighting that because I think it's an interesting and cool point, but it's also a nice example of where you can have sort of high specificity for specific things you're grabbing here, and then you dump it onto a delivery truck. Glutamate can deliver this as sort of a generic delivery truck into the urea cycle. Okay, that maybe was a little bit of a random detail on this. There's lots of details that I'm just not going to go into this in the interest of time. But the big takeaway is that we're investing a lot of stuff to detoxify urea or ammonia, sorry. And the detoxification involves taking an ammonia and turning it into a non-toxic urea. The final urea is a Franken molecule. So it has this right here, this amide is derived from the carbamyl phosphate. So this comes from whatever, say, amino acid. Go back here, right? That's the part that goes up through this whole complicated thing and then gets dumped into the urea cycle. We get one nitrogen from an aspartate. We've got HCO3 minus is the carbon, and then oxygen's coming from water, right? So this is this molecule that we've glued together to make the nitrogen less toxic. This molecule does not have the ridiculous pKa of an amine or ammonium. So this is now a non-toxic, safe to be delivered. And so this urea cycle happens in the liver, then urea gets dumped out into your bloodstream where it then ends up going to be disposed of in the kidneys and urine. And if you just look at nitrogen-carrying molecules in your bloodstream, urea is one of the higher concentration molecules. Okay. So that's what we do with the nitrogen side. We do this urea cycle thing. What do we do with the amino acid carbons? And it turns out there's basically two things that we do. To help set this up, I want to remind you of a metabolic pathway I've talked about in previous videos, hopefully you've encountered, and it is the idea of gluconeogenesis. And so gluconeogenesis is the process that our liver cells, our liver runs, for example, and what it does is it takes metabolic precursors like pyruvate and oxaloacetate, and it runs glycolysis effectively in reverse to make glucose. And this is really important in us when we, for example, are under starvation conditions. We have very low blood glucose. This is the way the liver can synthesize glucose from scratch to keep our blood glucose concentrations high. And you may recall, or maybe you should be reminded or told here, that the inputs to this, pyruvate and oxaloacetate, often come from amino acids, right? So an amino acid can be converted into pyruvate or oxaloacetate, and then used for gluconeogenesis. Okay, cool. Turns out that amino acids are basically broken into two major classes. The first are the ketogenic amino acids, where their side chains, when they're broken down, enter the central carbon metabolism as acetyl-CoA. I've used in the past this idea of different networks of transit networks, right? So they're coming in as part of the acetyl-CoA transit network, which is adjacent to and touches, but is separate from the glucose network, the glucose network. Glucogenic amino acids tend to come in or do come in inside of the glucose network. So you can think about this as, in the transit network analogy, this one comes in on a subway line. This one comes in on the regional train line, and they can be interconverted at a couple of points maybe, but these are very, very metabolically distinct blocks of reactions. Okay, so let's talk about what it means chemically that something's ketogenic versus glucogenic, and then we'll talk about what that matters for here. So, for example, tryptophan is ketogenic, or at least part of it's ketogenic. And the way that we know that is it starts out as tryptophan, and then if you walk through all of its breakdown pathways, and there's a lot of reactions, it ends up as this molecule acetoacetate, which is basically acetyl-CoA glued together. So this is on its way to becoming acetyl-CoA. The problem for this, or the constraint this adds to the metabolism for this, is that any acetyl carbons that come in as acetyl-CoA are lost to carbon dioxide in the citric acid cycle. So remember our tryptophan here, it made our acetoacetate, that becomes acetyl-CoA. And acetyl-CoA, quick reminder, has 2 carbons in it. That does not look like a carbon. There's 2 carbons in an acetyl-CoA. When that enters the citric acid cycle, it's going to start going around this way. And what we see here is 1, 2 carbon dioxides get popped out by the time it gets over here. So both of the carbons from acetyl-CoA have been lost before we get back to oxaloacetate. Now let's imagine that we take that oxaloacetate and we start making glucose out of it. We say we don't care. Well, now we're depleting one of the key intermediates of the citric acid cycle, and we're going to cause the citric acid cycle to stop. So at a net level, acetyl-CoA just isn't enough to drive the citric acid cycle. What that means is the ketogenic amino acids are not sufficient to be used for biosynthetic intermediates to make things like glucose. They just can't. So that means that basically these can be used for energy, but not anabolism. And so let's imagine that you are on a strict keto diet, you have no carbohydrates, and then you decide you're going to be totally crazy and you're only going to eat lysine over here. It's a strictly ketogenic amino acid. That'll eventually be disastrous, right? Because your liver's going to be working really hard to keep your blood sugar up. Where is it going to be sourcing that blood sugar from? It's going to be sourcing it from amino acids. If the only amino acid that's around is ketogenic, it's not going to have those amino acids. So it's really important, particularly when we go into conditions with low calories where we're needing to take amino acids and turn them into sugars, that we have a mixture, or that we have a complement of the other type of amino acids, these glucogenic amino acids. So what is a glucogenic amino acid? It's one that comes in somewhere besides acetyl-CoA, basically. So histidine, for example, is glucogenic. Hits up here. There's all these steps it can take. Ultimately, it ends up as alpha-ketoglutarate. We've seen alpha-ketoglutarate before, right? That's part of the citric acid cycle. But it comes in after one of the carbon dioxides is lost, right? So it comes in here. And so if I'm dumping in lots of molecules here of alpha-ketoglutarate, I go here, I lose a carbon. So this is a 5-carbon molecule, and we end up as a 4-carbon molecule at succinyl-CoA. But net, I'm still putting carbons into the system, right? So alpha-ketoglutarate can go up and form glucose without fully depleting the citric acid cycle of its intermediates, right? We're still dumping mass in. So if we're pulling mass out, as long as we balance the input outputs, we can use alpha-ketoglutarate as a source to, say, make glucose. What this means is glucogenic amino acids can be used for both energy and anabolism, unlike the ketogenics, which can just be used for energy. Okay, and then finally, actually, a lot of amino acids are mixed amino acids. They're both glucogenic and ketogenic. And what I mean by what basically that means is that the breakdown of that molecule leads to both products being made. So, for example, threonine can go into acetyl-CoA, one half of threonine goes on to acetylaldehyde and acetyl-CoA. The other half of threonine actually goes in as glycine, which ultimately ends up as pyruvate. So breaking down threonine gives you both acetyl-CoA and glucose, so it's both ketogenic and glucogenic. Okay, so that's kind of what I want to cover with amino acid metabolism. I quickly summarize these here, and then we'll be done. So amino acids are synthesized from a few central carbon intermediates and other amino acids. And you can sort of think of them as these branching pathways where they start with these central carbon intermediates and go out and to become more and more complicated. We can't make the essential amino acids. We have to consume these from the diet. Those are, we can synthesize 10 of them. The other 10 are complicated synthesis pathways that we outsource to our organisms that we eat. Then we talk about the breakdown side of amino acids. We said that there's 2 processes. You've got to break down the nitrogen side and the carbon side. On the nitrogen side, we use the urea cycle to detoxify ammonia. And on the carbon side, there's basically 2 classes of amino acids we generally worry about. We worry about glucogenic amino acids, which can be used for gluconeogenesis, and ketogenic amino acids, which cannot, which ends up being important when we're thinking about sort of dietary restriction and when we're thinking about starvation conditions. So. Okay, that is all I have on amino acid metabolism. Hopefully that was helpful. Look for a video here soon about nucleic acid metabolism, and I will talk to you later.